Good afternoon, everyone. My name is Frank Hollinger, and I am the president of Sphaera Pharma. We are the U.S. subsidiary of Sphaera Pharma Private Limited, which is a Singapore headquartered company. We've developed a remarkable drug modification technology. Very simply, it's a new chemical way of making prodrugs of molecules with the ability to repurpose, reposition, reprofile, and rescue drugs and drug candidates. You got to ask yourself, what's the problem we're really trying to solve? Well, whether you realize it or not, the drugs that you take that are out on the market, over more than half of them are actually less than ideal. There's problems with them. Not so much that it's going to cause a problem for you to take them, but enough to say that these could actually be better. They have suboptimal potency, they have suboptimal dosing. There's typically some form of undesirable toxicity with many drugs that are out on the market, and many of these drugs are either having patents that are expiring or they've already expired. So from a business perspective, that's not a good thing, because you've got a patented drug, you obviously demand a premium. If you've got better properties, you also demand a premium. So what's the need in the industry? Well, we need the drugs to work better, to make them safer, and from a business perspective, to be able to manage the drug life cycle. What this really translates to is you need to be able to generate new chemical IP around your existing drugs. So at Sphaera Pharma, over the past 5 years, we have developed a prodrug technology, a chemical way of modifying compounds, and I'll show you in a slide or two exactly what that really means in a graphic way. That allows you to take a drug, add a chemical piece to it, so it gives you novel IP, so it gives you a proprietary edge, that's capable of really overcoming a variety of the drug delivery problems that I just described. We have enabled this on over 30 different drugs, and based on the class of drugs and the variety of therapeutic areas, we believe that it can be applied to nearly any drug that is put our way. The outcomes of applying this approach is improved solubility, improved delivery of the drug, that is the ability to get the drug not only into your body, but to get it to go where you need it to go. And the combination of those two things translate typically to improved safety. And as I mentioned, one of the really big outcomes is the ability to generate patentable new chemical entities. And that's where the business comes in. So I'm going to go into probably some geek speak for a minute, so please bear with me. For those who don't know, prodrugs are very simply what we call a parent drug, basically a drug that you would take, that has had a chemical modification, a chemical change to it, that allows you to make your body more accepting of that particular compound, or allow you to formulate it, basically put it into a pill or a solution that is more accepting to your body. So the traditional prodrug approach, which would be, let's say, some competition of ours, would typically, the typical drug won't be able to go through the intestinal lumen. So typically you'll take a drug, right? Anybody here take Aleve or aspirin or any kind of drug like that, any kind of oral medication? Well, it goes into your stomach and it gets passed through your intestines. And some drugs, they just won't pass through. So a very common thing to do is to make a prodrug. You add on a small chemical change to it, and that combination allows it to pass through the intestine, go into the blood, and when it's in the blood, it undergoes a biochemical transformation and releases basically the drug itself and releases this piece that you added on. Now, the traditional prodrug approach, these pieces sometimes can cause some of these side effects that you may encounter. Our approach is actually a bit different. So if you've got drugs that are only semi-permeable, that'll only go through a little bit, or don't go through very well, let's say in the stomach, but can transport through as it's going along the passage of the intestine, we add on a piece to the drug, hence the prodrug component, that once it encounters the intestinal lumen, basically the lining of your intestines, there's enzymes at that surface that will cleave this piece off, release the drug so that it can slowly go through the intestines, and release this what I picture as a star here, as a moiety that will actually just then traverse down the rest of the intestine. This piece is by the Food and Drug Administration called GRAS, and that's generally regarded as safe. So we typically put on things there that are very simple to come off, and also extremely safe and known to be safe by the FDA. It gives us a tremendous advantage, but we'll hit on a little bit later. So then the drug will slowly get released as it's going through your intestine, and now you've got in the blood, just as you had before, but in a more effective way, the final drug. So what does this ultimately translate to? Where's the business around this? Well, we can apply this technology to drugs that are already out on the market or drugs that are being considered for development, either in the pipeline, getting ready to go into human trials, or even those drugs that are in human trials. There's 4 main ways that we could that you can apply this. One is to repurpose the drug, repurpose a drug that's already out there. For example, taking a drug that's known for oncology and repurpose it for anti-infective purposes. And this has been, usually you have to discover that there's this alternate use. We've actually licensed this technology to a company down in Georgia for this exact purpose. Another component is to be able to reposition the drug. For example, you may want to change the route of administration from intravenous to oral, or vice versa. Maybe you've got an oral drug that you would like to make intravenous. A very common example of this is the first, people here have probably used Tylenol or may have given Tylenol to your children. Well, till recently, it's only been available in an oral dosage form. Recently, by making it an IV dosage form, it's actually being used with higher order narcotics, opioids, post-surgery, and all of a sudden you've got a whole new avenue for these drugs. The ability to also reprofile the drug allows you to change the way that the compound is distributed in the body. And lastly, the ability to rescue drugs or drug candidates. This is the ability when you've got a drug that's at the end of the development pathway where you've spent millions and millions of dollars, and you need to get extra solubility or an additional property that allows it to get into the body so that you can actually see the efficacy. This approach will allow you to accomplish that. Now, before I go on, I'm going to tell you about a couple programs specifically that we're focusing on from the many that we've enabled the technology with. But I want to assure you that the leadership team that we have advancing this technology and leading this company is veterans in the industry, both in drug discovery, development, and in the business world. Myself, I've been in drug discovery for over 20 years and have a proven track record in U.S. and international collaborations leading to a number of IND clinical candidates. The Chief Scientific Officer, Sandeep Dugar, has over 25 years of drug discovery experience. He's a serial entrepreneur, as I am. He's actually one of the few people in the world that could actually lay claim to actually co-inventing two drugs, not just one, but 2 drugs on the market. And I'd also like to note that he was also the driving force behind Sios's kinase platform that got bought out by Johnson & Johnson for over $900 million. 2 other key principals on our leadership team are George Schreiner and Richard Johnson, both with over 30 years of drug discovery and development experience, and their specific areas of expertise is that they're both clinicians, and their expertise is in the renal disease area and cardiology. I want to assure you also that we have a tremendous scientific advisory board that we tap into throughout our entire process in terms of the technology development and the programs that we look at and select for drug development. Among this team, we have partners and collaborators that have collectively put over 300 drugs into clinical trials, and they've collectively put in, actually gotten over 100 drugs approved through the FDA. Their skill sets range from early stage chemistry, process chemistry, marketing, as well as manufacturing of drugs, and very importantly, the regulatory components of getting drugs into the clinic. So I want to tell you very briefly, as time is short, about 2 applications that we are specifically focusing on and have spent the past year and a half evolving to identify candidates. One is a repurposed drug for it's a repurposed generic drug that used to be used for angina, and we identified a new use for it, for chronic kidney disease. And the second one is oncology, in the area of oncology, and it's about reprofiling and repositioning paclitaxel. So for those who don't know, chronic kidney disease is, in the U.S., is a tremendous problem. It's the 8th leading cause of death in the United States, affecting over 31 million patients. Over 140,000 new patients are diagnosed with kidney disease every year. The disease progresses through a series of stages, from stage 1, where it essentially starts getting detected, all the way through stage 4, where patients basically spend 3 days a week, roughly 4 to 5 hours a day, hooked up to a machine, as you see here, and they're on hemodialysis. There's other forms of dialysis, but basically this is the fate that it's just a matter of time before they get to. The step after this is either organ transplant or stay connected to the machine. So there's tremendous unmet need in this space, because the current therapies currently only have the ability to slow the progression of chronic kidney disease. And what's remarkable is that 75% of the patients that respond to that treatment will actually start developing resistance after 1 to 2 years. At Sphaera, we have developed a prodrug, SPR 494, that has both improved and a preferred drug profile for chronic kidney disease. It has global composition of matter, so we basically have taken this generic drug, using our chemical modification, have now basically re-energized the patent life of this drug. And the reason we've been able to do this is because through our collaborators and our partners, we've discovered a new biological mechanism that this particular drug is involved in. It's specifically about mitochondrial protection and regeneration, or biogenesis. Now this is really critical because on the path of kidney disease, basically the kidney begins to fail. One of the core elements that happens is that the mitochondria basically lose their function and die. And so this drug has the ability to slow the progression by itself. We have also found that in combination with current frontline therapies that are blood pressure lowering agents, that we actually have the ability, because we're using 2 very different mechanisms. It'd be the equivalent of blocking the Autobahn and whatever major highway that you would use to basically avoid a crash on the Autobahn. Okay, it's really creating a unique opportunity to now halt the progression of the disease. Because of the new function that we've discovered about its ability to actually have mitochondrial biogenesis, there is the potential for regression of the disease. That is yet to be determined, proven. There's also a very unique opportunity that we have with this drug, in that it's been approved. Its discovery started over 20 years ago in Japan. It was taken from the Japanese company to Europe, and by the time they got to the United States, the patent life basically ran out, because they started with a company that went through a series of mergers, and this particular drug was never filed for approval here in the United States. So that's a unique opportunity for the United States, because there's absolutely no opportunity to be able to prescribe to off-label. And in the rest of Europe and Japan and the rest of the world, our modification basically creates a different profile so that you cannot prescribe just the general generic drug. So SPR 494 will be a first-in-class, excuse me, sorry about that. So SPR 494 will be a first-in-class therapy for chronic kidney disease. We also have, there's proof of concept that's been demonstrated on the generic drug. So one of the simple blood test biomarkers is proteinuria levels. It's one of the things they use to early define the disease. In post-analysis of patients that were on this generic drug, they found that proteinuria levels were lower. This is based on a UK published data. Our own internal studies have demonstrated that it synergizes with blood pressure lowering agents in a predictive animal model that was used to actually approve the only now first-line therapy for halting or slowing the progression of kidney disease. And the generic drug has an established human safety and toxicity history. All drugs have challenges. Here, you never know until you get into clinic whether you're going to actually work or not. However, we've got enough data to give us confidence and to reduce the risk that we will have high efficacy in clinic, and we've mitigated the risk by leveraging and demonstrating our efficacy of this particular compound in models used to approve other frontline therapies. The other potential challenge here is regulatory, because we would need to pull in safety data from the past 20 years from Japan and Europe. The markets are huge. My time is short. Let's just say these are huge markets, multi-billion-dollar markets that are game changers for the patients. Because you can just imagine, if you're diagnosed with the disease, somewhere in your future, your future has a machine attached to your arm. And so if there's any hope to be able to stop that, that's going to make a game changer in this space. Since time is short, I'm going to very quickly go through the paclitaxel situation. Very high level overview. Paclitaxel is the #1 oncology drug used in the world. It's currently generic. There's a number of issues with it. Key among them is its toxicity. One of the components of its toxicity is what it's actually formulated in, what they use to solubilize it in the IV formulation. We've come up with a new way by applying our platform technology to this compound, we've been able to increase the solubility of paclitaxel, and we've actually been able to formulate it now in saline as opposed to chromophore, which is the excipient that causes a side toxicity for dosing of paclitaxel. So the key outcome from making our particular prodrug is that we've eliminated the need for the chromophore in the formulation. It's been able to open up and widen the safety window, and it also has opened up the avenue to be able to provide a new route of administration. Currently, it's only available intravenously. We have the ability to actually dose it IP, and we've got some animal tests that demonstrate its efficacy in that space. The market, again, for paclitaxel itself is huge. It's still over $1 billion a year. A modern formulation that's coupled with albumin, it's a protein that's meant to solubilize paclitaxel, is got a market of about $250 million a year, but that's an extremely expensive formulation compared to the generic paclitaxel. We believe we'd be very competitive in the paclitaxel market space. So for both types of these drug opportunity programs, the development strategy for us is very straightforward. We've done this before. We need to do our IND enabling studies, carry out clinical studies, basically phase 1 in to study safety and tolerability in healthy volunteers, followed by phase 1b studies, which would study the safety, tolerability, and preliminary efficacy in patients. For the paclitaxel case, we would just be carrying out phase 1b studies for obvious reasons. Concurrent with all of these activities, we'd be doing our business development activity. What I show here is an illustration of it. I think I still have another minute and a half, based on when I started. I'll finish up very quickly. So this is a typical timeline. We expect within a 2 to 3 year period to be able to get our phase 1b data, and for each particular drug at a cost of between $2.5 and $3 million. Our exit strategy would be a licensing opportunity after phase 1 data to a medium or large pharma company with conservative deal estimates about $100 million, with milestones staged over time and royalties on the back end. And if we don't get reasonable terms, we could carry the program forward to a phase 2 study, which would increase the valuation of the program, but obviously also have a higher deal exit. There's many opportunities that we can experience for partnering the technology, from straight out licenses to joint development programs where we would help the partner advance the program. We're seeking $6 million. We expect it to be tranched based on success milestones in advancing the programs. And the investment will cover our business development, preclinical and clinical development, as well as operating expenses for the company. I covered all the things, all the topics, and I know I'm out of time, so if there's, do I have time for one question, or?
So when do you envision the phase 1 starting?
It's contingent on funding.
Contingent on funding.
Yes.
Thank you.
So within approximately a year to year and a half after funding, we would envision starting clinical trials. Based on, and this is based on our past experience. We actually have 2 partners that, one we've out-licensed the technology to, that's actually done it, used it for repurposing an oncology compound for anti-infectives, and another joint venture spin-off that is pursuing an anesthetic that's basically a reformulation play.
Thank you.
And thank you very much for your time. I appreciate it.